Hi, I'm Matt Rivera, and Chris and I are going to talk to you about the monomers of the cytoskeleton. The cytoskeleton is a complex network of highly structured protein filaments and tubules that give a cell structural integrity, organization of cellular functions and components, and the ability to move and change shape, among several other functions. Each type of molecule composing the cytoskeleton has unique subunits which associate in different ways to form greater structures. In this diagram, you can see the different subunits and larger structures that each different type of structure forms in the cytoskeleton. And this is a fluorescence microscopy image of a staining of the different parts of the cytoskeleton. So I will first talk about the intermediate filaments. In general, the function of intermediate filaments is to help the cell withstand mechanical forces and protect genetic material. Their structure consists of a hierarchy of associated components. The most basic component are their protein monomers, which have a rod-shaped alpha helical domain, as you can see here. The type of monomer determines the function of the intermediate filament. For example, filaments made of keratin form structures in epithelial cells. Note that, unlike microtubules and actin filaments, these proteins are not globular. This rod-like shape maximizes intermolecular forces between monomers. Also, note that intermediate filament monomers have two different terminal domains with no structure, which helps differentiate the directionality of the monomers, as you will see. And in this diagram, you can see the two different terminal ends that have a different shape. These monomers coil together to make helical dimers, as shown here. These in turn associate to form antiparallel tetramers, in which both ends are the same, unlike actin filaments and microtubules, which have distinct ends. Here you can see the ends which are the same, which forms a sort of symmetrical structure. The tetramers combine in units of 8 tetramers, which associate with other groups of 8 tetramers to form a long, dense, rope-like structure, seen here. This structure can be connected to other intermediate filaments or to other portions of the cytoskeleton via a protein called pectin that physically links those structures together.
Hi BIOS 41, my name is Chris Reyes and I'm here to talk to you today about actin filaments and microtubules. Actin filaments, also known as microfilaments, help support the cell membrane. The actin filaments associate to form strands in the shape of a double helix just like DNA, as shown above. The actin filaments have a globular monomer, which means that it's round, and not rod-like like that of intermediate filaments. Some of the many functions of actin filaments include providing tracks for movement, aiding in muscle contractions, helping in endocytosis, giving pathways for intracellular transport, cytokinesis. During mitosis, intracellular organelles are transported by motor proteins to the daughter cells along actin cables, which of course are made of actin filaments, and the actin filaments also help form cleavage furrows, which help the cell divide during mitosis, as shown above. Moving forward from actin filaments, we have microtubules, which are hollow cylinders in shape. All of the monomers have the same orientation, unlike the antiparallel intermediate filaments. The subunit is tubulin dimer, which is composed of alpha tubulin and beta tubulin, and each tubulin is a spherical protein. The monomer is polar in structure, having a positive and a negative end. The monomers come together in different ways to form different polymers. For example, if 9 triplets of microtubules form a centriole forms, which is star-shaped as shown here. If 9 doublets of microtubules form, cilia and flagella, which are wheel-shaped, can form.